a hypothetical scenario that I want you to paint in your minds. Imagine that you are a researcher working with a wildlife species, or perhaps you are approached by a manager that works with a wildlife species. Could be a bird or a mammal or anything else, really, a plant even. And you have some questions, basic ones. You want to look at the genetic diversity in the population, you want to know the genetic structure, and you want to run some parentage analysis. Standard stuff, right? So you very diligently go into getting SNPs, perhaps from DART sequencing or even RAD sequencing, and you start applying your filters, right? You filter for SNP quality, read depth, missing data, minor allele count or frequency, and individual quality, removing individuals with a lot of missing data. And then you get some results, right? You look at heterozygosity and you find that females are significantly less heterozygous than males. You also find that the population appears to be divided into subgroups, and you find 91 parent-offspring relationships. Now, what if I told you that these results are wrong? That in reality, there was no differences in the heterozygosity between the sexes, that there was not a division in two subgroups in the population, and that you could have found 104 parent-offspring relationships, and not only 91. What did you miss? What went wrong? Well, it turns out that when you were doing all your filtering, you forgot a very important step, and that is to filter out sex-linked loci. And this is a very important step because many population genetic analyses assume that you are using autosomal loci. So the goal for me today and for this session is to convince you that sex-linked loci matter and that you should pay attention to them. And at the end of the talk, you can tell me, Diana, I am convinced, Diana, I am not, and then we can talk a bit more. So, what are sex-linked loci? Let's dive into it. Sex-linked loci are most of the time found in XY system. Females have two X's, males have XY, and for the ZW, females are the ones with ZW, males have two Z chromosomes. For our friends working with amphibians, fish, and reptiles, some can have XY, some can have XW, some can have different types. And I recently learned that actually cannabis, the fun plant, has XY sex determination systems. So there you go, plants too. And there are three main types of sex-linked loci. Well, there's actually three types of sex-linked loci. I will use the ZW system to illustrate them. The first type is the ZW, and those are the ones found only in the W chromosome. So when sequenced and genotype, they are called or present in females, but absent in males, right? So if we plot the proportion of heterozygous females versus the proportion of heterozygous males in which a locus was called, W-linked loci are here, down in this corner. So present in premature females, but absent in males, allowing for some sequencing errors. On the contrary, autosomal loci should be around here, with basically no difference in call rate between the sexes. The next type of sex-linked loci is the Z-linked, that, as you probably guessed, are the ones found in the Z chromosome. For these loci, females have only one allele. So when genotyped, they appear as if they were homozygous. In reality, they are hemizygous because they have one allele, but when you see it in your data set, they look homozygous. Males, on the other hand, can be homozygous or heterozygous. So again, if we plot the proportion of heterozygous females versus the proportion of heterozygous males, Z-linked loci would fall here, with no females being heterozygous, right? And again, autosomal loci would be mostly here, with no differences in heterozygosity between the sexes. The third type of sex-linked loci is the gametolog. And these are interesting ones. They are the ones found in homologous regions of the Z and the W chromosome. And for these ones, there is one allele that is found exclusively in the W chromosome. Therefore, males are always homozygous, and females can be homozygous or heterozygous. And now, this is exactly the same, just in the opposite direction for the XY sex determination system. Do not blink, do not blink, do not, sorry. So the gametalogs are also here, right? With no males being heterozygous. As I was saying, this is exactly the same for the XY sex determination system, just the opposite direction. Do not blink. There. Exactly the same, just opposite. It's flipped. All right, so now we know about sex-linked loci. What can you do with them? Well, you can either remove them or keep them. Removing them is a very important thing because in this way you can facilitate unbiased population genetic analysis. How would you go about, though, removing these sex-linked loci? They are a bit tricky, aren't they? Like, proportions change, sexes, what do you do? So here, for example, we have the dataset, real dataset of a mammal. Can you please give me any ideas on how would you go about removing sex-linked loci? We know that these ones over here are probably autosomal. We know that these ones over here are probably sex-linked. How would you remove them? Any ideas? Anything goes. No dumb answers. Whatever. Yes, yes, correct, yes. Yes, if maybe you realized in the previous versions of Dart-R, the approach that the function was using was to setting thresholds, right? So you would kind of say, okay, below this threshold, I consider that sex-linked loci, same in the other axis. That's how you remove them. Can you think, though, of some disadvantages of this approach? Sorry? Yes, you have mainly two disadvantages. One is that you lose some resolution when you start talking with low frequencies, right? So this part over here. And also, how about that point over there? This tiny one here. Is it sex-linked? Is it not? Where do you put the threshold? How do you justify it? Tricky. So in the new version that we have in our package called Dart-R sex-linked, we designed a function that identifies them automatically. Choose a threshold, we already did it for you. And we will explain how. The function is called GL filter sex-linked. And it uses the property of autosomal loci of having this roughly one-to-one relationship in call rate and proportion of heterozygous individuals between the sexes. So it applies a statistical test to identify loci that significantly deviate from it. The function works in two stages. Sorry. First, for each single locus, it performs a Fisher test to check whether call rate is independent from sex. And if it is not independent, then it means that it is sex-linked. So this is the example. We have here the contingency table of one locus. And in here you can see that if you're a male, you're very likely to have this locus called. And if you're a female, you are very likely to have it missing. For this test, you would have a significant relation. Yeah, you would reject that the sex and the called rate are independent, and therefore the function assigns it as sex-linked. Your data set would go, and this is a real data set, looking from this before using the function to that, and from this to that. FYI, the function also gives you these plots, so you can see the effect of the function with your own eyes. Little note here. There is another type it is not really another type of sex-linked loci, but for the purposes of this function, we call them sex-biased. And it's this funny curve, this funny aggregate of loci here that are not behaving like the W-linked or the gametologs or the Z-linked that we talked about. But we found out that these ones are Z-linked loci. So this is data for a ZW system. And these loci over here are more cold in males because they are Z-linked, because males have 2 Z chromosomes. Females have only 1, right? So if we look at the call rate in female, this is 0.5. The call rate in males goes something around 0.7. And that's simply because, given that males have 2 Z chromosomes, it's more likely to be called, right? To be sequenced and then to be called. And I am very, very proud to announce that this function has an average of 98.8% accuracy in correctly identifying autosomal loci. And this for us was super important because we wanted you to trust that whatever you use as autosomal loci down the line will actually be so. Right. I am selling to you the function, but maybe you say, okay, Diana, is this really necessary? Like, can I just omit this? Really? Well, we tested this, and the paper is already published. We used actually 3 datasets, but I will tell you about 2 datasets of 2 beloved Australian birds. We used the dataset of the yellow-tufted honeyeater and of the eastern yellow robin. And for each dataset, we applied 2 filtering regimes. So one incorrect filtering regime, which we only applied standard filters, and one correct filtering regime, in which we filter out sex-linked loci first, right? So sloppy one and a good one. And we asked 2 questions. First, how good are standard filters at removing sex-linked loci? So in the first case, right, in the sloppy regime, do I get rid of sex-linked loci? And in the second one, if I don't get rid of them properly, what happens to population genetic analysis? And this is what we found. Follow me, I will explain this. At the top, we have the initial data set that starts with 100% of all the sex-linked loci that are present in our data. And at the end is the final data set. Happily, we found that standard filters, redepth, and loci missing data are very good at removing W-linked loci. They just don't make it to the final data set. But bad news. That is not the case for Z-linked and gametologs. You can apply all these filters, and at the end, you will still have over 50% of Z-linked loci and over 1/3 of the gametologs in your final data set. This is a data set that you will use to run genetic analysis. And the same for eastern yellow robin. For eastern yellow robin, it was even higher. Over 70% remained. And these loci actually represent 5.7% of all the loci that you have in yellow-tufted honeyeater are sex-linked. And for the eastern yellow robin, they represented at 7.8%. This is not a negligible percentage. So, take-home message number 1, standard filters suck at removing all sex-linked loci, okay? They don't do the job very well. Now, we used our sloppily filtered data set to run genetic analysis, and we found this. We found that males are 5% more heterozygous than females. And if we were to believe those results, we could think that perhaps females are philopatric, or perhaps inbred males are dying, so that there's inbreeding depression. We also found with the sloppily filtered data set that there was this division in 2 groups for each sampling location that we had, which maybe could have made us think of 2 cryptic sympatric species. And we also found 91 parent-offspring relationships, which could be used, and it's very common, to use them in the calculation of extra pair paternity rates, or to guide captive breeding, or to guide translocations. But all these results that were spurious disappeared or improved in the case of the parent-offspring, when we properly removed sex-linked loci with our function. Are you scared yet? Good. Therefore, take-home message number 2, sex-linked loci can F up your population genetic analysis. So, take-home message number 1, which one was it? Standard filters suck at removing sex-linked loci. Take-home message number 2, sex-linked loci can F up population genetic analysis. Remove them. Cool. So we talked about removing sex-linked loci. The second thing you can do with them is to actually use them. And one of the most common applications for that is to sex the samples for which you don't know the sex. So just as we did, we used the known sex individuals to identify sex-linked loci. You can do the opposite. Once you know sex-linked loci, you can use them to sex the rest of your samples. For that, we created another function that is called gl-infer-sex. And what happens most of the time, what we have seen people reporting, is that they use one type of sex-linked loci. Perhaps they identified gametologs, and then they use those gametologs to sex the individuals they didn't know the sex for, right? So for example, if it's a gametolog, I have an individual that appears to be always heterozygous, so it's very likely to be male. The beauty of this function is that it uses all the sex-linked loci that you have, that you found with the previous function. And I think it's very cool, yes?
I've been stuck on the gametologs because I'm like way back where you said that, so like males, so ZZ males can be heterozygous or homozygous, but for some reason males can't be heterozygous for gametologs, and I don't understand what's different in this figure.
The gametologs, you mean?
Yeah, so what's the difference between males in, so if you're looking at the homozygous sex, so in this XX,
sorry, yeah,
what's the difference in the homogametic sex between green and orange?
Okay, so for gametologs, you have one allele that is found only in the Y chromosome, right?
Okay.
So these males could be homozygous or heterozygous. But females can only be homozygous because the funny allele is only found always here.
So then why can, in the X-linked, in the XX, you're saying it can be heterozygous?
Here.
So like looking at the XX, what's different between green and orange?
Sorry, I'm not getting the question.
I think it's not that cannot be, it's less likely to be heterozygote because recombination and linkage in the Y chromosome. So the Y chromosome can mutate and will never pass to the X when the X can be the same allele most of the time.
Exactly.
So it's more likely to be heterozygote in the males, less likely to be in the females.
Exactly, yes, right. It's not always the case that they are homozygous or heterozygous. Yes. This also depends on the age of the sex chromosomes. When you have old sex chromosomes, it is more fixed, the differences are fixed. When you have younger sex chromosomes, this varies a lot, and that's why it's tricky. So, let me continue. Yes, so the function uses all the types of sex-linked loci that you find. And if you want to feel very fancy, this function actually uses machine learning algorithms to classify them as as females or males. So you can feel very fancy when using this function that you are using machine learning. And the function gives you as output a table that looks like this. For each type of sex-linked loci, it gives you the sex that the individual has according to that type of sex-linked loci. If all of them agree, you have a very nice, very clean sex, and if they don't, you will see them marked with an asterisk. We defined this function with the specific and explicit intent that to all the asterisks you see, there should be a person, this is you, checking the counts and checking the evidence and checking the numbers of W-linked, etc., for you to make the call of what sex it is, okay? So don't go just removing asterisk and just keeping the sex that is there. You have to make the call. It is also the time in which you can go back and check your samples, check your notes of fieldwork, etc. You need to make the call. And the beauty of this function is that because you're using all the available evidence, your sex assignments are way more robust. Right, so now let's talk numbers, because science is not always perfect, fieldwork fails, etc., etc. The minimum amount of known sex individuals that you need is 30 known sex individuals, 15 males and 15 females. The genotype object can have many, many individuals, but at least 30 should have sex. You use those ones as input for filter sex linked, and those ones will help you identify some sex-linked loci. Not all of them, because with only 30 individuals, you don't have the statistical power to find them all, but you should find some. You use those ones then as input to infer sex, to sex the rest of your samples, and then you do a last round of filter sex linked to find the rest of your sex-linked loci. Does this make sense? Yeah? Cool. And then you are ready to go to do quality filtering, right? All the other filters that Renee told us about. Now, question. Why are we recommending to first remove sex-linked loci and then go into quality filters? I will give you a clue. You know these ones, you are familiar with these ones. You have the call rate by locus, call rate by individual, and read depth by locus. Why do you think you need to remove sex-linked loci first? Any ideas? Yeah, you might remove individuals, like differentially between the sexes. Something else? It's pretty much part of the same thing. So for example, these poorly called loci, many of them are likely to be Y-linked. And over here, poorly called individuals are likely to be males because males only have one X chromosome, therefore, they have less chance of getting all the X loci called. And over here, these tiny ones, there will be many sex-linked loci that have a poor read depth simply because they don't exist in some individuals. So by removing sex-linked loci first, you guarantee that first, you won't unfairly remove individuals from one sex. And second, that downstream filters will actually remove bad quality loci. Here you will be removing Y-linked loci, and they are not bad quality, they are just sex-linked. You remove those ones, and then you evaluate quality. Make sense? Cool. Take-home message number 3. Remove sex-linked loci first. Also because if you remove first, missing for loci, you would be getting rid of Y-linked loci, etc., etc. So, take-home message number 1. Exactly. Standard filters suck at removing sex-linked loci. Take-home message number 2. Sex-linked loci may F up your population genetic analysis. Take-home message number 3. Remove sex-linked loci first. And now, just wrapping up the talk, you can use sex-linked loci to sex your samples, and there are many other applications. So you can study sex bias dispersal with that. Robin has done a lot of work on that, so there we go. And then you can also study the evolution of sex chromosomes, which Arthur is an expert in. So yes, I am an expert in you have sex-linked loci, you want to get rid of them. Florian, on the other hand, is you really want to identify sex-linked loci. How do I do that? Talk to Florian about those topics. And I will just leave you with this chapter in which Florian is a co-author in, a very good resource. And this is the paper in which we publish the rationale of all the functions. Questions?
Hi, yeah, it's really great advance to data R and a really great advance just generally. So great to see that. I'm just wondering, just a quick question. Do you draw upon the count data that data provide now routinely as part of their service? Or do you not require the counts to apply these functions?
The count of what?
The count data, you know, the read count data.
Ah, yes, read depth. It doesn't. It doesn't use read depth. It's just based on those two things, call rate and heterozygosity.
Okay, yeah. So it'll work for everyone's data sets, even if they generated them 18 months ago as well as now.
Exactly.
Okay, great,
exactly.
Thanks.
Diana?
Hi.
I'm going to ask some questions for the guys online.
Sure.
So one question was about if you have a species with more than 2 sex-linked chromosomes, can you still use the function? How is the filtering effectiveness going to be?
Yes, so Florian was telling me about a case in which there was a species in which you had both XY and ZW sex determination systems. Yeah, there was something funny going on there. Yeah, there was a recent fusion or something like that. So what you can do is actually test them both, right? You do one run in which you put the males and you say it is ZW, sex determination system, and it will find the ones that go according to that. And then you can run it again in the autosomal ones, in which then you declare XY, and then you identify those ones. Did I say it correctly? Well, anyway, you run it once with one system and the next one with the other system.
One more question from online. Is that okay?
Yes, sure.
Yeah, sure. There's a question about hermaphrodites. Are they unaffected?
Can you repeat that, sorry?
Hermaphrodites, so if you have a species that is hermaphroditic.
Hermaphroditic. That's a good question. So I think our philosophy has been, if in doubt, just check. You know, if you don't know anything about your sex determination system, try it with both. I mean, it's pretty obvious, right? If you see something that doesn't follow this rough one-to-one relationship, there's something sketchy going on there.
But wouldn't you have to know which ones are contributing to the male and which ones are contributing to the female to get that relationship? But if they're hermaphrodites, you wouldn't know. You'd just be a mess. It'd be all over, right?
Okay, yeah, fair. I'm not sure. I don't know. I think we will have to test it. Yeah.
I was just wondering whether there's a way to use this approach if you don't know like any of the sexes. Do you think you could look at the proportions, frequencies, and try to use it to assign sexes or find sex-linked loci kind of iteratively?
Yeah, not with this approach, because Fisher test is based on this contingency table in which you do know the sexes of some individuals. So not with this approach. There are other approaches. I don't know. I know of rat sex that uses the reads, but I think they also require to know sex of some samples at least. Yeah. We can think about it.
This is probably the stupidest question we've had all week, but given we're meant to apply this to what we're doing to all organisms, there are some organisms that have multiple mating types, which in other species we'd call sexes. Do you think you could extend what you're doing to something with multiple mating types?
Sorry, can you repeat the last part?
So some fungi have up to 30 different mating types, which are like our two sexes. Two sexes is boring, rare and strange. So do you think you could extend what you're doing to cope with organisms that have many, many mating types within the one species?
Well, I think it is doable. I guess I would first go about testing as it is a function, seeing what you find, and then the rationale behind it is still there, right? This one-to-one relationship. Surely we can find something, I guess, yeah. Like we can collaborate and see how would we need to tweak the functions to, yeah, to identify other things, yeah. But at the moment, give it a go. Let's see what you find.
Just had a question. I guess showing the birds, you had a sort of percentage of the loci that were linked, but does that vary a lot between different groups? And is it really more important in some groups than other groups?
Yes, that's a very good question. We had the eastern yellow robin and the yellow-tufted honeyeater. Yes, they are both passerines, but they are from like decently far away in the tree of the passerines. The interesting thing on eastern yellow robin is that they have neosex chromosomes. That's why that percentage is larger. What happened is that you had the sex chromosomes and they got stuck with the I think it's the 1A chromosome. So at the end you have like this very long neosex chromosome that just has a lot of loci. But what it's recently being found in birds, but I suppose it's being found also in other organisms, is that at the beginning we thought that these chromosomes were relatively conserved, but more and more we're finding rearrangements and fusions and things like that. So unless you have a genome that is chromosome level genome and that you have tested for sex chromosomes, I would again just go test it. It doesn't hurt, it's one step.
I might be able to elaborate on that a little bit. I guess you've got fugu, which is a fish which has its sex determining locus is one SNP, and then you've got mammals are fairly conserved, but even in mammals, rats have like 6 copies of SRY, and so you're going to find a lot more signal in a rat than you are in, say, an ordinary mammal. And then in Pagona dragon lizard, it's got its sex-linked. The only sex-linked sequence we can find in that with a lot of looking is on a repetitive element, a line sequence, and it's only 3 base pairs. And so you don't expect to, given we're all working on different model organisms, don't expect miracles to come out of your sex-linked marker thing, because it's very species dependent. Yeah. And so all the birds are probably fairly conserved. And also they can have very big Y chromosomes, but the non-recombining region, compared with the PAR region, can be very small. And the sex-linked markers are all in the non-recombining region, they're not in the para-autosomal region. So, every species is going to be different. And so, you need to know a bit about your species to generate an expectation.
I know, it's a bit cliche, but depends on the species.
How robust is this method if, say, 10% of your sex metadata is wrong? So like when the individuals were sexed, some of them it's not perfect.
Yeah, you can go back to the 15 known males and 15 known females. So ideally, if you have some individuals in which you are not super sure, remove them, because they might introduce some bias. So just keep the ones for which you are sure, and as long as they are 15 and 15, preferably more, at least 15, you should go well. Yeah. In doubt, just remove them. You know what I mean, right? Because then
I know what you mean, but if you don't have certainties for your sexes, right? Okay. So like working with data that someone else has collected, they haven't always sexed correctly, but I don't know which ones they were certain about, which they weren't,
Yeah.
So how robust is it if some of them are just wrong? Will it still pick up the patterns?
Just to quickly answer on that, so I've run well, sharks is really hard to sex as well. So I've got data which had up to 30% misidentified sexes, and they work. But as long as you're not at 50%, and then it's just purely random, you will still be able to pick up. Like I said, the gametologs and the Y-linked or the W-linked markers are easier to find. The heterozygosity markers will be a bit more tricky if you're the wrong sex.
Here,
Yeah.
can I just very quick, that could be one cool example where you can do a power analysis using data. You just mix some of the sexes and see how long can you find that, and then you get at least some confidence that even there is some in your data. That would be my advice. Yeah.
Alternative, what people sometimes do is they use other methods like PCR methods, sexing methods, for just a few individuals, and then you use those ones. And then you have your sex-linked loci, and then you can sex anything else you want. Yeah. Any other question? Good. So.